Today's topic is hemoglobin. We'll discuss about synthesis of hemoglobin, functions of hemoglobin, breakdown of hemoglobin, its variants, and hemoglobinopathies. First of all, what is hemoglobin? It is basically a quaternary protein. Okay? It has 4 subunits and it is present in the cytoplasm of red blood cells. Okay, it is a conjugated protein formed by conjugation of two things. What are those two things? Heme and the globin part. Okay, as you can see in this diagram, heme part, it is attached to the globin chains, alpha and the beta chains, and heme has iron. Okay, so it is helical shaped polypeptide molecule. Now about the normal values. Normal values, these are different in newborn children, males and females, as you can see here. So there is a reason to this, why the value is more in newborn as compared to children, why the value is more in males as compared to females. So these reasoning based questions, they'll be discussed once we'll briefly complete about hemoglobin. Okay, about the structure of hemoglobin, as we had already discussed, there are 4 subunits. Okay, each hemoglobin has 4 subunits, and heme, it is an iron containing porphyrin complex called as iron protoporphyrin 9. Okay, as you can see in this diagram, iron, it is present in the center in ferrous form, protoporphyrin ring plus iron as ferrous. Okay, and porphyrin nucleus, it consists of 4 pyrrole rings. Here you can see it consists of 4 pyrrole rings named as 1, 2, 3, 4, and these are linked to each other by the methine bridges. The carbon atoms of these methine bridges, these are named as alpha, beta, gamma and delta. You can see in this figure, and 8 side chains are attached to the pyrrole rings. Here you can see the side chains, methyl, vinyl and propionyl. Again, a representation of structure of hemoglobin. Okay. Now iron, it is attached to the pyrrole rings by nitrogen atom, you can see in this figure. And oxygen, it is loosely attached to ferrous at the 6th covalent bond. So each molecule of hemoglobin, we can say it consists of 4 ferrous, and each ferrous, that is linked to 1 oxygen molecule. So in short, we can say that one molecule of hemoglobin, it combines with 4 molecules or 8 atoms of oxygen. Again, in representation of structure of hemoglobin, you can see the numbering of pyrrole rings, the bridges named as alpha to delta, and the side chains 1 to 8. Now about the synthesis of hemoglobin. Heme part, it is mainly synthesized in mitochondria and cytoplasm of the immature RBC. Globin part, it comes from ribosomes. Heme and globin combine to form hemoglobin. Now once it is synthesized, it has to get broken down also. So what is the fate? As it is composed of heme and globin, again it will be broken down to heme and globin only by the macrophages. Heme part composed of iron and porphyrin. Iron, it will go to the iron store to get reused, okay, or get stored. And the porphyrin part, it will be converted to biliverdin first, which is a green colored compound. That will be converted to yellow colored bilirubin, and it will be excreted out by liver and bile. The globin part, proteinaceous part, it will go to the amino acid pool. Now about the functions of hemoglobin. Hemoglobin, it carries the transport function. It transports oxygen from lungs to the tissues. We had already discussed that one molecule of hemoglobin binds with 4 molecules of oxygen or 8 atoms of oxygen. Binding of oxygen to the hemoglobin, it is called as oxygenation. Don't get confused with oxidation. Oxidation is different. Here it is oxygenation, formation of oxyhemoglobin. Now this oxyhemoglobin, it is of bright red in color. It can be simply written as HbO2. Okay. Now binding of one oxygen molecule, it increases the affinity of hemoglobin towards the further oxygen molecule to such an extent that the affinity for the fourth oxygen, it is 500 times as compared to the first oxygen molecule. And 1 gram of hemoglobin, it contains around 1.34 ml of oxygen when it is fully saturated. So this point is very, very important, that the affinity of fourth oxygen molecule is 500 times than that of first oxygen molecule. Okay. Reason is attachment of one oxygen molecule increases the affinity for the next subsequent oxygen molecule. Second function is the transport of carbon dioxide from tissues towards the lungs. Okay. And removal of oxygen from the hemoglobin, it leads to formation of deoxyhemoglobin, and the color is dark red. Now this deoxyhemoglobin, it forms carbamino compounds very quickly as compared to oxyhemoglobin. So we can say that venous blood, it is more suitable for the transport of carbon dioxide from tissues to the lungs. Third action is the buffer action. It is 6 times more capable than plasma proteins in the maintenance of pH. Now about the types of hemoglobin. Most common is HbA, adult hemoglobin. Okay, it can be represented as alpha 2, beta 2, means it has 2 alpha and 2 beta globin chains. Next is, and very few percentages given to HbA2. It has 2 alpha and 2 delta chains. Next is HbF, fetal hemoglobin. It has 2 alpha and 2 gamma chains. Now this fetal hemoglobin, it is more resistant towards alkalis as compared to the adult hemoglobin, and the affinity for oxygen is more as compared to adult hemoglobin. So commonly students are asked questions, what are the differences between adult hemoglobin and fetal hemoglobin? So you can simply write fetal hemoglobin, it is more resistant towards alkalis, more oxygen affinity is there, but the lifespan is less. Lifespan is 120 days for adult hemoglobin, whereas 80 days for the fetal hemoglobin. Now about the derivatives of hemoglobin. So hemoglobin, specifically the heme portion, it binds with several ligands and form the derivatives of hemoglobin. Here are few examples. Oxyhemoglobin, it is of bright red in color and it is formed when hemoglobin binds with oxygen. Next is your reduced hemoglobin. When oxygen is released to the tissues, the hemoglobin, it is reduced or deoxyhemoglobin. Next one is carbaminohemoglobin. Binding of carbon dioxide to hemoglobin forms a darker red colored compound called as carbaminohemoglobin. Carboxyhemoglobin, binding of carbon monoxide at the 6th covalent bond forms cherry red colored compound carboxyhemoglobin. Methemoglobin, whenever the blood is exposed to the oxidizing agents, the ferrous form, it is converted to the ferric form and there is formation of methemoglobin, it is of dark brown in color. Then glycosylated hemoglobin. Non-enzymatic attachment. Non-enzymatic word is very important here. Non-enzymatic attachment of glucose to the terminal valine in each beta chain. It forms glycosylated hemoglobin. Okay. And it is used as a marker for the polycontrolled diabetes mellitus for the estimation of long-term glycemic control in these patients. Sulfhemoglobin. Sulfur, when combines with the heme part of hemoglobin, forms a greenish derivative of hemoglobin. It cannot carry oxygen. Now a brief description about hemoglobinopathies. Sickle cell anemia and thalassemia. Details of these two, they'll be covered under anemia. Now I'll be just briefly describing what are hemoglobinopathies. Whenever there is any defect in the globin moiety, not in the heme part, it is called as hemoglobinopathy. Heme is normal. There is problem at the amino acid part. Sickle cell anemia. It is a form of your hemolytic anemia where glutamic acid, it is replaced by valine at the 6th position of beta globin chain. It occurs in around 10 to 20% of Negroes and have origin from the African black population. You can see here the sickling of the RBCs. As the name suggests, sickle cell anemia. Anemia is there and RBCs, they become sickle shepherd. Now there are main two forms of this disease. Sickle cell anemia, homozygous trait is there, it is autosomal recessive. Whereas sickle cell trait, it is heterozygous condition and Mendelian dominant trait. It is inherited as Mendelian dominant. Okay. Now, this carrier trait, it offers protection against P. falciparum malaria. Don't get confused. P. falciparum protection, it is only for the patients who are suffering from sickle cell trait, not sickle cell anemia. Here is the inheritance pattern for sickle cell anemia and sickle cell trait. You can see here. Which one is heterozygous, which one is homozygous. Sickle cell anemia is homozygous, sickle cell trait is heterozygous condition. Next one is thalassemia. It is an autosomal recessive condition and there is mutations in the genes that reduce the synthesis of alpha and the beta globin chains. It confers protection against malaria. So basically there are two types of thalassemias depending upon the absence of particular globin chains. Alpha thalassemia, again subdivided into 4 types. Hemoglobin Barrett's hydrops vitalis, there is complete absence of all the 4 alpha chains. HbH disease, there is absence of 3 alpha chains. Alpha thalassemia trait 1, 2 chains are present, 2 are deleted. Alpha thalassemia trait carrier stage, only 1 alpha chain is deleted, 3 are present. Beta thalassemia, there is beta thalassemia major, also called as Cooley's anemia or Mediterranean anemia. There is complete absence of the beta globin chain. Next category is of beta thalassemia intermediate degree of severity is there. Beta thalassemia minor, there is not absence of globin chains, there is only reduced synthesis of the beta globin chains. So out of thalassemia minor and thalassemia major, which one is more common, less common? Okay. Details of this thalassemia and the sickle cell anemia, they'll be covered when we'll discuss about anemias. So thank you for now. We had covered hemoglobin and in the next lecture, we'll be discussing about red blood cells. Thank you.